All right, we're going to give this a try here. I want to walk you through the next section of our textbook and our notes looking at the microscopic anatomy of skeletal muscle. It's really important that we get this part down now because as we transition to the neuromuscular junction tomorrow, we're going to need both the microscopic anatomy known well, the neuromuscular junction known well, all leading up to the big process coming up here on Wednesday, which will be the sliding filament theory of muscle contraction. So a lot of information coming at you here. I'll do my best to walk you through it. Oh, try to share this. All right. So looking at our notes here, when you see sarcolemma is no different than the cell membrane or the plasma membrane of a normal cell. When we use the prefix sarco, it just helps us remember that we're talking about muscle. Myofibrils are the long organelles inside of the muscle. We also call them sometimes myofilaments. Later we'll give them specific names, actin and myosin. They're the protein filaments that allow our muscle to contract and relax. The last one on here, the sarcoplasmic reticulum, our specialized smooth endoplasmic reticulum inside of the muscle cells. A reminder back to chapter 2, the endoplasmic reticulum's job is to package things up and ship them into and out of the cell in certain places. So we'll talk about that in greater detail when we get to the sliding filament theory of muscle contraction later this week. So to give you an idea of what it looks like, this here is an individual muscle cell, or as the book guys that goes back and forth, they just call them muscle fibers. Inside of the individual muscle cell are a whole bunch of the myofibrils that make up the protein filaments of the muscle cell. So when we get to looking at skeletal muscle underneath the microscope, you probably remember that it is striated, and by that it kind of had these banded patterns of kind of dark and light areas within it. The striations are because of the myofibrils that are inside there. The I band is the light band, and it contains only thin filaments. Specifically, its name is actin. The A band is a darker band. It's the entire length of the thick filament, and we call that myosin. So this is a picture that you're going to want to get to know really well. It's figure 6.3 in your book. It's found on page 188. There's a lot of things happening inside of here that we absolutely need to know. If I could break down a couple of things really that we want to get to here. The blue lines in between here are the thin filaments. Okay, we call that actin. And the dark red here are your thick filaments, which are called myosin. Now, the space in between the thin filaments is called the H zone. Now, we're going to talk about the sliding filament theory of muscle contraction later this week. It's the H zone that's going to shrink and disappear when the thick and the thin filaments are playing a role with each other as the muscle is contracting. The I band is the place that we call in between the thick filaments. And right down the middle of the I band is what's called the Z disc. The Z disc is what kind of anchors down the individual thin actin filaments. So there's a lot going on here. The M line, which is located here, goes right down the middle of the H zone, and that's the portion where the muscle actually contracts. Again, this will all probably make more sense as we start putting these things together later this week. But this is going to be one of those things that you're going to want to ingrain in your brain and get to know the thin actin filaments and the thick myosin filaments as we go. So again, a couple other terms here that are important. The sarcomere is the contractile unit of a muscle fiber. So the distance between the two Z discs right here, that's called a sarcomere. So a couple things when we talk about the organization of the sarcomere, the myofilaments, there's the thick ones, which are myosin, and the thin ones, which are actin. And we'll talk about how those two work here in a lot more detail tomorrow. So I know this is kind of where we're going to end for today, but there's a lot that happens between these thick and thin filaments in order to allow our muscles to contract and relax. First off, the myosin filaments are composed of proteins. They have binding sites for the enzyme ATP synthase. Now, if you remember back to 10th grade bio, ATP synthase is the place where ATP is made. ATP is the chief energy source for cells. And guess what? In order to contract and relax, our muscles need energy. So the binding sites for the enzyme ATP synthase is going to be a really important part of the contracting of the muscle cells. The myosin filaments have these big old heads on them, and they're going to be the things that actually do the contractile twitch of the muscle cell. And we'll talk about the heads of the myosin in much more detail tomorrow. Thin filaments are actin. They're composed of proteins as well. They're thin, and they are going to be the portion of the muscle contraction that actually gets moved, and that's going to be done by the thick filaments later on. As we said earlier, the thin filaments are anchored to the Z disc, and the Z disc is the anchors of the contractile unit of the muscle cell. So don't be afraid to rewatch this video. Send me any emails, or if you have any questions. We'll try to get a quiz and some workout tomorrow morning over this information. All right, take care.